do the complex inheritance Punnett squares. You need to answer number 1, so explain the difference between incomplete and codominance. You go ahead and do that. Look at the notes for the answer for that. Here we're going to go through the Punnett squares. So for the first step, we're going to make our key. In some chickens, the gene for feather color is controlled by codominance. Remember, codominance means they're both being expressed. The allele for black is capital B. The allele for white is capital W. The heterozygous phenotype known as erminette, which is a black and white spotted chicken. So let's make our key. Capital B is going to be our black chicken. Capital W is going to be our white chicken. Notice we're not using the same letters anymore. And we're going to have a capital B, capital W, that's our erminette, which is black and white spotted. When we go to do our cross, we have to read the problem. 2 erminette chickens were crossed. So we have to look at the key, erminette. It says erminette is capital B, capital W. Capital B, capital W, crossed with capital B, capital W. Remember, when we fill in the Punnett square, only one letter goes in each box. This represents each gamete. This is a parent gamete. So this could be a sperm, this could be a sperm. This could be one egg, this could be one egg. Then we fill it in, dropping down the B, sliding across the B. Drop down the W, slide across the B. We always keep it in alphabetical order. Drop down the B, slide over the W. And here we go. So for our genotypes, we start in this top left box. We've got capital B. There is 1 out of 4 boxes, so 1 out of 4, or we could say it's 25%. Then we go to the next box, capital B, capital W. Look how many boxes have that. That's 1, 2. So we have 2 out of 4, or 50%. And then our last box is our capital W, and we've got 1 out of 4, or 25%. For the phenotypes, I want to know what's it going to look like. So this first box, we're going to go back up to our key, capital B, that's a black chicken. So black, we have 1 out of 4, or 25%. Then we have our capital B, capital W. Capital B, capital W, that's our erminette. So we're going to say erminette, 2 out of 4, 50%. There's 2 boxes. Then we have our capital W, that's our white chicken. So white, 1 out of 4, or 25%. Moving on to number 3. We're going to use the same key. So it's the same problem, same information. We're just going to do a different cross. So I'm actually just going to copy the key. Paste that in. Okay. When we go to look for our cross, look at the problem. It says a black chicken and a white chicken are crossed. So black, we have our key, capital B, and a white chicken, capital W. Okay. So we're going to put 1 letter in each box. When we go to fill it in, we're going to find that every box has a capital B, capital W. So what this means is if these 2 chickens make offspring, every single offspring is going to have this genotype, and every single offspring will be erminette. So when we go to do our genotypes, so there's only 1. So BW, we've got 4 out of 4, or 100%. The phenotype BW, if you look back up at the key, is erminette. So we say erminette, 4 out of 4, or 100%. Now we're doing another problem, different information, so we can't use the same key. We're going to make a new key. So cows can either be red, white, or roan. Roan is a red and white spotted cow. So let's do our key. So we're going to use capital R. That's going to be red. We're going to use capital W. That's going to be white. And then we're going to do capital R, capital W. That's our roan, which is our red and white cow. For the cross, What offspring are expected from mating a roan bull, right? So roan, capital R, capital W, and a roan cow. So again, capital R, capital W. Fill in the Punnett square. Keep it in alphabetical order. When we go to do our genotypes, start in this top left corner box. We've got capital R, 1 out of 4, or 25%. Our next genotype is going to be capital R, capital W. There are 2 boxes, so 2 out of 4, or 50%. And then we have our last box, capital W, 1 out of 4, or 25%. For my phenotypes, I'm going to go back to the first box, capital R, capital R. Capital R is going to be red. So we've got red, 1 out of 4, or 25%. Our capital R, capital W, according to our key, that's our roan. So we've got 2 out of 4, or 50%. And then our last box is the capital W, which would be white. And we've got 1 out of 4, or 25%. All right, moving on to incomplete dominance. So incomplete dominance, we're going to see a blending of the 2 traits. Instead of seeing both of them expressed, we're going to see it like mixing paint. So red and white will make pink. In snapdragons, flower color is controlled by incomplete dominance. The 2 alleles are red, so let's start our key. We've got red and white, so capital R is going to be red. Capital W is going to be white, and then the heterozygous genotype expressed as pink. So capital R, capital W is going to be pink. A pink flowered plant is crossed with a white flowered plant. So look at your key. Pink is going to be capital R, capital W, crossed with a white, capital W. So if we put our pink flower here and our white flower here, now we fill it in, keep the letters in alphabetical order. Nope. Start at the top left box. Our first genotype is capital R, capital W. We have 1, 2 out of 4, or 50%. Our next genotype is going to be capital W. There are 2 out of 4, or 50%. For our phenotypes, we need to look at what color they are. So capital R, capital W, our key says that that is pink. There are 2 out of 4, or 50%. And then the capital W, our key says that that is white. 2 out of 4, or 50%. What this means is if a pink flower is crossed with a white flower, there's a 50% chance they're going to make a pink flower and a 50% chance they're going to make a white flower. Number 6, in birds, bluebirds. So let's go right to our key. Capital B is blue. And white birds, capital W, that's white. They're homozygous. The heterozygous bird is a bluish gray, capital B, capital W. So we put bluish gray. What results of a blue bird? So when we go to do our cross, blue, capital B, crossed with a bluish gray bird. Our key says that's a capital B, capital W. Fill in the Punnett square. For our genotypes, first genotype is capital B. We have 2 out of 4, or 50%. And then our next genotype is capital B, capital W. There are also 2, so 2 out of 4, or 50%. For the phenotypes, look at the key. So capital B, that's going to be blue. There are 2 of them, 2 out of 4, or 50%. And then capital B, capital W is going to be our bluish gray. 2 out of 4 or 50%. Okay. Now for the blood typing. Blood typing, here is your key. You don't need to make a key. It's already here for you. Homozygous B allele. All you got to do is look here, find where it says homozygous B. So homozygous dominant type B right here. So we're going to put capital B, capital B. Heterozygous for the A allele. So heterozygous type A right there. So A, lowercase o. Type O, there's only one choice. Type A and had a type O parent. Okay, so type A but had a type O parent means that it had to get an O from one of the parents. So it has to be this one, A, O. And then type AB, there's only one choice for AB. AB. Draw a Punnett square showing all the possible blood types for the offspring produced by a type O mother and a type AB father. So type O, we need to know the genotype. Genotype for type O is going to be 2 lowercase o's crossed with AB. The genotype for AB is just AB. So we put O, O, A, B. Again, keep it in alphabetical order. Okay, so when we go to do our genotypes, we've got blood type, we've got genotype AO, AO, 2 out of 4 or 50%. And then we have BO, 2 out of 4 or 50%. For the phenotypes, we're just going to write the blood type. So we're just going to write type A, that's our AO, 2 out of 4 or 50%. And then we have type B, 2 out of 4 or 50%. All right, for this last problem, we need to figure out if the parents possibly had their babies switched. And this is actually something that happens. It happens often. That's why babies and parents have the same wristband with a coded key on it. It's got a scanner on it, so that way they can match it to the baby and the parent. So they think their baby was switched. Mother has type O. So again, the genotype for type O is 2 little O's. And the father has blood type AB. So the baby has blood type B. Use a Punnett square to determine if the baby was switched. So we've got O, O, and then A, B. So AO, AO, BO, oh, not a 0, and BO. So when we look at the genotypes, we've got AO, 2 out of 4, or 50%. And then we've got BO, 2 out of 4, or 50%. For the phenotypes, again, we're just going to write the blood type. So we've got type A, 2 out of 4, or 50%. And then we've got type B, 2 out of 4 or 50%. So use the Punnett square to determine if the baby was switched. We would say baby might not be switched because it is possible to have a baby with blood type B. Okay, there's your answer.